Okay, then how do you grow more complex eukaryotic cells? Cells like our cells, right? Mammalian cells are good examples. Before we talk about the growth medium of those cells, let's talk about the cell culture of those mammalian cells. For example, there are important terminology you need to understand. So when you take those cells from their body, the animal's body, you can probably take up those cells through a biopsy, right? If you go to a doctor could do a biopsy on kind of suspicious tissue type, right? And those cells can grow inside of a tissue culture medium for a while. When you do that, we call that a primary cell culture. Those are the cells that are just taken up from living body, and they can be maintained for several generations. Okay, those are primary cell cultures. And those cells, like I said, since they can live continuously for a few generations, you can subculture or pass. Means what? When cells grow and they will expand and they will run out of surface they can stick to, and they run out of nutrients, then you will have to move those cells to a different fresh medium. That's what subculturing or passing is. You will get to do this in your cell culture class. Okay, but these cells, the primary cells, cannot live long enough. They will die up after a few days, maybe a week or so, because that in vitro, the test tube, is not the natural condition where they are used to, right? So they will die out eventually. So primary cells cannot live for a long time. However, there are certain cells that can live long time inside of a test tube. Okay, they're just like your cancer cells. We call those transformed cells because through a bunch of different mutation, those cells taken up the ability to live forever outside of a cell, outside of body, and inside of a test tube. Those are transformed cells. Okay, established cells, another term. Transformed cells or established cells. So most of those cell culture cells that you will maintain to, make API, those are the transformed cells. They will live continuously without losing their integrity. Okay. Understand what primary cells are, how they are different from maybe established or transformed cells. Transformed cells are kind of mutated cells. They can live forever. Primary cells cannot live forever. And then when cells leave, a certain period of time, they have to move from one medium to another because they will run out of space and nutrients. That's called passing or subculturing. Okay. And then what kind of media would allow continuous growth of mammalian cells like CHO cells? Okay, what kind of things you should provide the cells with? Now think about your body condition. Okay, what kind of condition your body cells would need? Your body has a lot of blood, which is full of nutrients and all the necessary ingredients, right? So you can grow cells in something called serum. Okay, serum is a liquid that is isolated from your blood. Okay, if you remove blood cells from your blood, that is a serum. Okay, because this has bunch of necessary proteins, polypeptide, amino acid, growth factors, and all those good things like lipids, carbohydrates, salt, hormones, and vitamins. So you can grow cells on serum, okay, without the blood cells. Okay, one rich medium is called serum. Okay. Another important components in the cell culture medium, you need to provide some inorganic salts. Okay? So salt is important to maintain that all those isotonic condition, of course. Okay, and then some amino acids are required because your cells need to build a lot of proteins, and the building blocks for proteins are amino acids. You need to provide those. And energy source, again, carbohydrates, simple sugar, glucose should be added. So glucose, sugar will be broken down from your mitochondria, and they will release energy that's required for the cells. So all these things are important. Okay. And then what else is important? The pH adjustment is important, right? Like the proteins and biological molecules, the cells should maintain the integrity of the pH as well. Then how would you adjust the pH of the cell culture if you're growing a mammalian cell? Okay, you can adjust the pH by using these two factors. One is sodium bicarbonate. Its chemical equation is this, NaHCO3. Another is the CO2 gas, carbon dioxide. Okay, so if the pH of your solution, your cell culture medium, became acidic because sugar is broken down continuously, and often the byproduct of the cellular respiration are the acid, like lactic acid and things like that. So the pH of your medium can become acidic. Okay, under that condition, you can add sodium bicarbonate into your cell medium. Okay, if that happens, your sodium bicarbonate will become HCO3 minus, which will bind with H pluses. So it'll take up H plus ions, which will make acidic medium, okay, from your growth medium. So by doing so, it's going to raise the pH of your growth medium. So it'll bring back to the neutral medium. All right. What if it's the other way around? What if your growth medium is too basic? Then what's going to happen? Then, of course, that's not a good condition for the cells. Under that condition, you can pump in carbon dioxide gas. CO2 gas can be pumped into your growth medium. If that happens, CO2 plus H2O will make this compound H2CO3, which will become, H plus and HCO3 minus. So by doing so, it will release a lot of H pluses, which will bring down the pH to the kind of acidic area. So by using either sodium bicarbonate or CO2 gas, you can adjust the pH. Okay, if the pH is too low, you can add sodium bicarbonate. If the pH is too high, you can pump in CO2 gas. Okay, and then normally mammalian growth medium has some kind of pH indicator. If it's not growing in the bioreactor, which has a pH probe already embedded in it, you could use a pH indicator, which changes color from maybe magenta to maybe yellow if pH drops down too much. Those are good indicator for the pH value of the medium. Okay.